I've seen many extremely interesting discoveries in academia which nobody ever developed. Let's say you start on the level 1 and you are fantastic. Go to level 2, you're fantastic. You go to level 3, you're fantastic. You go to level 4, you're crap. That's how the industry works.
Hello and welcome to the 3DCC, 3D Cell Culture conversation platform, wherein we are talking and conversing with the people who are leading and doing all the young innovations in the field of healthcare and 3D cell culture especially. And today I am pleased to be joined by Dr. Marcin, founder and CEO of Real Research and the pioneer in 3D cell culture and biomedical technologies. He is the creator of LifeGel, an innovative 3D cell culture platform for which is designed particularly to bridge the fundamental research between the real world biomedical and the industrial applications. And also, he has built interdisciplinary teams who are working on the global partnerships, advanced scalable 3D cell culture technologies that are shaping the future of translational research. So, it's my pleasure to welcome you on this podcast, sir.
My pleasure. Thank you for having me.
Before we jump into the scientific conversation that we're gonna have, let me know what has been like, how has been the journey for you in this particular domain so far?
Yeah. So it's a roller coaster.
Okay.
Yeah. So I think we will have we can expand it, during the discussion. But for us, it was really like me and my co-founder. We were we were working actually on a side project,
Okay.
and that was during my PhD. And we came up with the hydrogel as something we needed.
Okay.
And that was a very good thing for us. But then we thought whether we should, with time, we realized that there was much more value than just one or two publications.
Exactly.
And it was just a lot of time that we had to put just to make it all reasonably repeatable.
Yes.
And with that, we just thought, well, we could equally just start a company and doing that. And then we had really one crucial experiment that was a huge struggle. But after probably about a year, it actually worked very well. And then we decided to start a company.
And that's how LifeGel came into the
Yes,
picture.
exactly.
That's
Exactly.
wonderful. So, from your perspective, what distinguishes the true research platform from the pioneering but limited laboratory tool?
I think I like this question very much because I think the only difference is the devotion of the researchers.
Okay.
I have seen many extremely interesting discoveries in academia which nobody ever developed.
Yes.
And they all are just a publication somewhere.
And how do you think does an academic research differs now that you are also into entrepreneurship after you
Yeah.
discover something
Yeah.
going through the academia?
Yeah.
How much difference have you seen in the workflow that we have in academia and in the industry?
It is very different.
Oh, really?
It's a very different approach. It's like in academia, it's like Formula One. You just want to be the first one. The rest doesn't matter.
Okay.
If you do it once, you win the global award for whatever, and you're the
Yes.
best. Obviously, those top professors do it many times, and industry is different. Industry is like the bus. You just it has to be repeatable.
Yeah.
Every day, the same bus, the same time. You want to be at
Exactly.
work at the same time. If the delays are low,
Yeah.
industry has to make something. It's like from the scientific perspective, the industry is much more rudimentary, but the value it brings it's just on another level. You still can complain about products that come from the industry because nothing
Yes.
nothing is perfect, but the requirements to make something in the industry it's just a different world.
And coming back to the 3D cell culture
Right.
and all the work around revolving around this,
Yeah.
if you had to explain to someone in a layman language what 3D cell culture is
Yeah.
or what organoids and
Yeah.
all the innovations coming out of this,
Yeah.
what are these?
To be honest, I think it's a very easy and extremely difficult question at once.
Okay, both.
Both, because it's if you know, like, to make it to the easy part of the question is like we are three-dimensional.
Yes.
You are three-dimensional. I am three-dimensional. And if you think that you can make any research using flat surface, it's extremely intuitive that it's just the wrong way. So this is the easy explanation.
Okay.
But the difficult explanation is when you go into details, what's happening there, how, because you can have a 3D model that's wonderful and you can have a 3D model that will mislead you. And that's the big question, how to validate that, how to make that repeatable. And going back to your previous question about the difference between industry and academia, that's really the whole story about making, it's not about making the best ever possible model once, it's about making reliable, good enough model, but million times. So really the explanation of what is this 3D cell culture, it's really an attempt to grow something in the lab that will behave as very, very small part of human body.
Okay, like similar, mimicking.
Yes,
basically.
exactly, but very limited. So a tumor mimics a tumor, not even the whole tumor, but a small subset of tumor. Like cornea, it's just mimicking the function of cornea. Like liver, you can have 20 liver models, and some of them will be good for this disease or the other disease, or healthy liver to test toxicity.
Yes.
So those 3D models can be extremely good at one thing. And then this thing has to be very well defined.
Okay. And when we connect the dots of building these 3D cell culture and organoids and everything to the real world impact, how does that happen? Like these are used for the personalized medicines or therapeutics? How?
I think most of the applications are still ahead of us.
Okay.
At this moment, the applications are limited to drug discovery, chemicals testing, a little bit of regenerative medicine, and real world applications. That would be it. But the long term applications sound like science fiction. Sometimes it's really amazing. Like you've probably heard about the brain computer interface, or you obviously like advanced regenerative medicine, like replacing organs, like giving, like for example, curing genetic diseases by implanting modified cells.
Yeah.
Those things are slowly happening, but they are at this moment extremely expensive.
Okay.
And okay, if they work, they can do miracles, but I literally know 2 or 3 of them, and each one of them costs $2 or $3 million. So,
and when we talk about 3D technologies that we have,
Yeah.
they most of the time, as you already mentioned, the important thing is reproducibility, repeatability of your own
Exactly.
product,
Exactly.
the innovation that you're doing, also scalability and all.
Yeah.
So how does Life Gel been designed to support the standardization and the broader adoption to all of these things?
Yeah. So we see ourselves like one instrument in a huge orchestra.
Okay.
It's we are focusing on being able to deliver one product that's just extremely repeatable. Once we define which type of gel we do, that has to be like McDonald's. Just wherever you go, whether you're in India, US, or whatever, it has to be the
Yes.
same. And that's our goal. And if you look at any cell culture, you just need 3 ingredients: cells, gels, and cell culture media. So we do gels. And I think that's a very good spot because there are many companies who do quite good cell culture media. I think there is still some improvement that the industry would benefit from, but
Yes.
it is good enough. In terms of cells, there are so many different sources of cells.
Exactly.
The bigger problem is which one to choose, not that there is actually almost nothing you couldn't find.
Oh.
That's the good stuff. And in terms of gels, we have one major competitor who everybody complains about them, but still they have like 70% of the market. So yes, so you know what I'm talking about. And the thing is that we are really looking forward to changing the paradigm, especially in those new approach methodologies, because when you do something new,
Yes.
you don't have to use the old methods, and that's where
Definitely.
we are aiming at.
Yes, and when we talk about innovation, something that also comes in the picture later is
Oh yes.
the regulatory compliance,
Yeah.
because we cannot just ignore that. It
Oh,
also becomes a
the-
very important,
Exactly.
integral part of the whole innovation and everything. So, what are your perspectives on the regulation compliance in
Yeah.
the field of organized 3D cell culture and everything?
Yeah. That's the you couldn't find a better topic. That's the way to go. That's and to be honest, what your question between the question about the difference between academia and industry,
Yes.
it's actually very good question on the same line because it would be academia, industry, regulation.
Yeah.
Because regulation is even more strict than industry.
Yes.
And I think that's because without regulation, nothing will work. Just it's simply going to be illegal. First of all, there will be no pressure to do those things in a way that then industry would be able to apply this to real products. And luckily the governments, not all of them, but many of them see the need, and there are very clear attempts. I wouldn't call them clear paths, but attempts that they know. Actually, there was here on the conference, I think
Yes.
the second speech was by one of your ministers.
Yeah.
And he was,
The
I liked the
India.
way, how open he was, that we can do it, but we're not the best at it. And, that was good that he listens probably to other people.
Yes.
And I think that's really the way to go, because once the governments make decisions about how the regulation would look like, then there will be much bigger incentives for the industry actually to apply those norms.
Yes.
And before that, it's much more risky for the industry to do
Yeah.
something.
I mean, something can work, but when we regulate the
Exactly.
thing, it works perfectly.
And
And-
that's also true, but I'm trying to touch a different point.
Okay.
You can come up with a solution, and if your solution works and it gets banned, you're done. That's it.
Yeah, exactly.
You can come up with a
Yeah.
perfect virus to transfect patients' samples, and then they can say, well, lentiviruses are okay, but adenoviruses are not, or the other way around. And yours was the wrong one. So
Yeah.
once they set the standards, let's say you can optimize towards the standard.
Exactly. And you also have been operating at the intersection of science and entrepreneurship. Yes.
Yes.
For
Yes.
several years. So, as I already had this question, I had asked you this question, the industry, academia,
Yeah.
how it works together. So, what is the kind of key in between this, that the industry academia collaboration is working out? What is the key for this?
Are you asking about the question about the like day-to-day work between academia and industry, or are you talking about the kind of going from academia to industry? Because I'm not
Okay.
sure.
So what I'm asking right now is that when an industry academia collaboration is taking place,
Yeah.
what kind of perspectives do you have out of this, that the benefits that you
Okay.
want out of this?
Yeah. So to be honest, for us, it's extremely well specified.
Okay.
It is. We have some collaborations with academia, and they are from the day one till the last day
Yeah.
just defined. So sometimes we need some expertise, and that's what we're looking for. There are other times where we just need some techniques that they
Yeah.
help us with. Sometimes we want to publish together, so we just divide the work
Yeah.
we need. This is like I think the biggest challenge in working with academia is just to find the right people. And if you just found the right people, it's
Yes.
just a pleasure, I would say, because
Yeah.
for publishing it is actually even more exciting because you can do the little bit more crazy stuff.
Yeah.
And then of course internally you worry later about standardizing that in your company, but at the
Yes.
beginning that is actually
But the foundation depends on what is the purpose
Exactly.
behind this collaboration.
100%, and
Yes.
the human interactions basically.
Definitely. And now that we talk about collaborations and all, what kind of advice you have for the young innovators and the young researchers who are working to, create some innovations for in the same domain of organ on chip, 3D
Yeah.
cell culture related, or human relevant,
Yeah.
animal free tests they are developing, and all these innovations, right? So, what kind
Yeah.
of advice you have for them? What should they do? What should they look after?
That's difficult.
Okay.
Because I can give you this very general answer, which I don't like.
Completely fine.
I don't like this general answer, to be honest, because I think it's very misleading. Because the general answer would be look for something you get excited with and
Yeah.
find a top lab that does it, and just do your best to get there. That would
Yeah.
be the general answer. And I think it works for some people.
Yes.
But since you're asking me for advice, I just give another perspective. There is one thing I wish younger people knew at the age of 25, maybe. And that's how the industry works. And I don't mean exactly, you don't need to know all the
Okay.
details, but understanding where different positions are. So,
Exactly.
like when I was finishing my studies, I basically thought, well, there are some people who are selling the products, so working just as a salesman, but a little bit more educated. Or you just do research, and
Yeah.
maybe you can do some research in industry. And that was extremely naive. Now I feel like I understood 2% of
Yeah.
the industry. So first of all, understanding how different levels of work
Yes.
actually work.
Yes.
So and people very often want to get the higher position, and I think like in corporations there is this phrase which I totally agree with. You are stuck on the first position you're not qualified for,
Okay.
because let's say you start on the level 1 and you are fantastic. You go to level 2, you're fantastic. You go to level 3, you're fantastic. You go to level 4, you're crap. Nobody will say, "Oh, I want my salary lower and I want to go back."
Yeah. They
No,
won't.
you will
Yeah.
not say that. You will stay at the
Exactly.
first position you're not really good for. And I think that's one of the problems. And then there are fantastic people who worked in the lab, who actually stopped working in the lab, moved to a little
Yeah.
bit more managerial position, and they openly say, "I don't like working with people." You
Now
see?
they want to come back.
Exactly,
Yeah.
exactly. So I think understanding of how the ladder actually works
Yes.
I think that would help many people in making
Definitely.
decisions. And once you understand that, you can think about what your future can be.
Exactly. Now, before wrapping up the whole conversation we just had, I have like three rapid fire questions for you. I just need your instinctive answer for those
Okay, I will
questions.
do my best.
Okay, do
Yeah.
your best. So, in one word, tell me the biggest challenge in 3D cell culture adoption is.
Trust.
Okay, great. In one word again, the future of biomedical research platform is.
Whatever will be legally accepted.
Okay.
Sorry, it's not one
Nice.
word, but
No, it's okay, but it's a really constructive answer, I have to say. It's really good answer.
yeah.
So, 3D cell culture technologies will
Yeah.
transform in a decade in the research domain by.
I really think, but transform doesn't mean end of story.
Yeah.
It will be very different because of AI.
Oh, yes.
Very different. I'm not even sure whether in every single aspect better.
Okay.
I think there will be some problems, and I think people will have to go back to different things.
Yes.
But the AI sometimes
Yeah.
and good models. Okay, AI can be extremely innovative, but what I think is really crucial is that AI learns on the data that we provide, and many data that people
Definitely.
provide is wrong.
Yes.
So AI will learn, we will make mistakes, we will face a wall, we will have to go back, and that will happen in many different directions. So some of the directions will be a wonderful breakthrough.
Yes.
Some of them will be a total failure.
Yeah.
And
but then we can learn from them.
Exactly.
Definitely.
That's why the 10 years is actually a very good timeline, because
Yeah.
many things will change.
Exactly. Additionally, I have one question. So, how do you see 3D cell culture platforms which are there integrating with the organoids, organon chips, and the NAMs, especially
Yes, yeah,
the new
yeah.
approaches in the coming next decade?
Okay, this is actually very, very. I think there is already an answer to this, because to get anything regulated, you need to find that specific question that your model answers.
Yes.
So all of those that you mentioned have to be defined to a very, very, very narrow line of the question. So what I would say is all of them, like they will be simply very big spectrum of methodologies
Okay.
that you will then apply. Probably with time, the number of them will narrow down, because some of them will be validated for more and more and more. So
Okay.
let's say you start with a liver model that, let's say healthy liver model that is good for testing, I don't know, urine-derived toxic substances. Let's say this.
Exactly. Yeah.
Then you let's say you have a company that works and provides this model, and you realize, okay, we can also add another layer of validation to our model to do 5 other things. And then with time, you realize that this model, it's not perfect for everything, but
Yeah.
instead of one, it can work for 50 different things. Not 500, but 50.
50.
So what I'm saying is, I think first, like now, I think now we are before the storm. After the storm, you will see a lot of chaos with
Yeah.
hundreds of different models, and then they will start to consolidate.
Yes. Then we can choose and then we can standardize them basically.
Yes, but I think the consolidation is crucial because after the storm you will be able to choose. But it's
Yes.
like going to a restaurant with 5,000 different things on the menu. It's extremely hard to choose, and then they have to consolidate to, I don't know how many. That's a total unknown to me. And then we will start to see something that is really scalable.
Yes.
Because some of those systems are also proprietary. So what happens if you have a company with 40 people and I don't know, not all of them, but let's say 10 of them decide to leave, another 10 retire, and you're left with half of your stuff. It's like, there is know-how that leaves your company, or, your 2 key scientists accidentally die in a motor crash. Things happen, and then what I'm saying with that is systems that will be used in the future have to be robust enough to be able to be done basically with a certain training anywhere. And most of the NAMs you can see, most of the best ones, or those that actually claim the most spectacular results, are extremely protected.
Yes. So this is how the NAMs should be designed basically.
Yeah. So there are many attempts
tackle.
for them to be scalable.
Yes.
But very few people can repeat those results.
Exactly.
You can always say they are not financed enough, nobody invested enough, and that could be true.
Yeah, obviously.
But eventually somebody has to be able to repeat that.
Exactly.
And without, sorry.
Yeah, I would have loved to have more conversation with you on this topic, but obviously we have some time constraint over here.
Yes.
But it was really wonderful talking to you because
My pleasure.
more than just being insightful, it was inspirational talking with you on this topic, like 3D cell culture and all this, as you have a wide experience, and it showed with your words over here. Thank you so much for giving us your time. Thank
My
you for
pleasure.
being here.